Okay, so welcome back to the day 2 of RNA-seq I Analysis. And today we're going to cover the second part of the workshop, which is going kind of more in-depth into the thick of the pipeline, which include reference genomes annotation. And then we have, we'll align the process reads we have processed yesterday to the reference genome. So in the case of the examples, we're using human genome. And then we want to tally per gene read count. So that's getting to the point where we have the gene, count table. And then that will be kind of the basis for all downstream analysis. And then we'll talk about the bulk RNA-seq pipeline, which, basically by that time, we should actually go through every element of it. So we just want to kind of give it a try using a mouse liver data. So that's for today. And then for tomorrow, we'll have kind of an overview of other aspects, including downloading public data. And we also want to kind of get into visualizing the data. We want to normalize it in a way that we want to make sure that, everything is done correctly. So let's dive into it. So for those who are taking for credit, there's going to be a quiz. So by the end of today, you should be able to complete the quiz because all the contents will be covered by then.
Is it due today?
It's due next week. Yeah, next Tuesday.
The quiz.
The quiz you should be able to complete the quizzes by the end of today because all the content taught today will be able to cover you.
But it's not due today.
It's due next Tuesday. Okay, so we're coming back to this kind of workflow. So we have gone through the first part where we get to, we process the reads, we understand, what's in them. How do we figure out, do we have to filter? How much do we have to filter? How to filter? We're using what tools? And today we're going to go a bit, deeper into the second part, which is after we have processed the raw read, now we want to align them to the reference genome. And we're going to use that, use the software STAR. So that is available on Hoffman too, and that's very convenient. So let's just go a bit deeper into sequencing alignment. So what do we actually do, right? You ask the question, people always talk about alignment as if it's, a given. But it's actually not a trivial process in terms of, it's quite a statistical problem. We have a pool of mRNAs. We have a lot of sequences coming out of, next gen machine. And you have all those fragments. You want to find out where they come from the genome. So that's a problem where you have to actually match the sequence to the reference genome. And that comes with a lot of uncertainty. For example, there are a lot of tandem repeats. You couldn't really find out where it's coming from, right? Because you can match the same sequence to multiple parts of the genome. So that's one scenario. Another scenario is if the fragment is too small, you cannot really resolve them well. Because for short sequences, there are a lot of chances that the random genome or random sequence can be, mapped to in the reference genome. So that's an issue that was pretty much the focus of the end of 20th century, where a lot of actually algorithms were developed to try to just tackle this specific problem, which is given a sequence, is there an efficient algorithm to match it to a reference genome, which is super long and complicated in terms of their structure. They're linear. That's kind of the idealized idea, but you have different chromosomes, when they cross, at the centrosome, there might be cross-linking. And then if there are kind of, deletion event happening, which happens in a lot of somatic genomes, you do exome sequencing, there are a lot of deletions, or you have mutations, you have a whole chunk missing, right? Given all those variations, can you still match it back to the reference genome efficiently? So that's pretty much the idea of the alignment process. And we have a lot of software to do that nowadays. So for us, it's less of a concern. So this is what it usually looks like. So we are looking at a genome browser view of a segment, which is on chromosome 19. And then we're looking at, a region within a gene, PDE6C. And it's typical to have a lot of those kind of exons in a sea of intergenic regions, like introns in between, or between genes, there's going to be a long strand of sequences that doesn't really have any annotation. So what happens for mRNA sequencing is that you actually have read that is only coming out from exons, right? Because mature mRNA would have all the introns being clipped, and then they are enjoined. So you end up having a lot of those fragments that span between different exons, and then you want to make sure that somehow they're kind of covering the space that you're interested in. And then for the others, you have maybe occasionally you have read coming from introns. So that happens if you have multiple splice isoforms. So that's possible as well. So let's just have a look at what the specifics of read sequence alignment. So the idea is you want to sort out the sequence from the pool of mRNAs to the specific origin from the genome, and then you want to determine is this a region that I know is for sure is coming from. If there's uncertainty, you'd better know, right? And then another thing is you want to make sure that we can do this efficiently. Sometimes it takes a long time. And I would say this is the bottleneck in terms of computation time. Alignment takes most time in terms of the entire pipeline processing. And we're going to try it today with the KV1's SC sample. And even with the cluster, I waited. It takes about half an hour. But you will see. This is relative the time that takes to process this step. And then, of course, mappings based on sequence similarities.
You said the fragments that were mapping to the reference genome, those are just exons, right?
The reference genome has everything, but annotation would actually make sure that you know where the exons are, right?
Yeah, because the thing that we're mapping to the reference are only exons, right? So there's no.
Not really. So the reference genome for the human, we know how big that is. It's 3 billion base pairs. So it's actually the entire sequence.
Okay.
You map it to the reference genome, but you want to find out where they are, right? That's where the annotation file comes in, and you will get to actually have both files. The original assembly, which is the reference genome itself, just raw sequences, and then the annotation files would actually mark specific regions in the reference genome to tell you, oh, this is this gene, and for this gene, we are looking at exon 1, 2, 3, or intron, or 5 prime, 3 prime UTR region. So the tallying step would actually make sure that we are counting the right region. Does it make sense?
Yeah.
Okay.
It has to align to every single chromosome of the reference genome.
Right. Yeah. So for each read, you have to find, somewhere. Right. And it's quite an exhaustive process. That's why it's, taking the most time. But with modern computers, it's relatively easy. Imagine the early days of Human Genome Project, even putting the contigs together takes forever. I don't know how long it took for the whole project, maybe 10 years.
So what happens to those sequences that, if they're too short, for example, and it's it could match in multiple regions, so they get thrown out, or? Right.
So we actually make sure that the sequences are long enough in the filtering step yesterday. So that kind of take care a lot of that concern.
Base pairs, it should be fine.
Yeah, usually, for 20, it's reasonable. But when we were dealing with the sample yesterday was 75 base pair for each read, right? So that's fairly long, right? For short ones, short ones that happens to be filtered and it got mapped, what happens is the mapping will come out with the best score. And we'll get into that to find out what, mapping was high quality, and then we will be to find out low qualities. And then when you are doing processing with the softwares, you actually be able to control that. So yeah, good questions. So for different genomes, let's have a look. So there are different browsers we can use. So one of the, maybe you have used this before, the UCSC genome browser. So that's the Santa Cruz maintained genome browser. So they have typical genomes that is readily loadable, humans, mouse, and then a couple of other non-standard genomes as well. So this is updated. So that means you can choose the versions that you want to work with. And there are kind of archives as well. You want a specific version. So currently for human, I think it's H38. That's the latest, and then for mouse is MM39. Yeah, you can look it up. Just Google UCSC genome browsers, and typically you have the the annotations. So for example, we're looking at this specific gene, and then here's the gene body. It has orientation. So in this case, it's reading from the right to the left. So depending on the strand, it can be on the positive strand, negative strand. So that's where the orientation comes from. And then you have those islands standing for exons. And in between, those are the introns. And then there are different annotations. You can annotate it with different other informations. Cannot read it from here. But what is useful for this browser is that you can load different additional modalities. For example, people have done epigenetic sequencing, looking at methylation, acetylation, other data, or you can look at transcription factor binding data. You can load into this one browser on top of your data, or alongside your data. So you would be able to kind of make inferences. For example, this region, I have a very abundant read. What could potentially be regulating it? Usually the acetylation data means that the whole region is acetylated, so it's actively transcribed. If it's methylated, it can be an enhancer region, or it can be a repressed region, etc.
So these are all information that you load in that.
Yeah, you can load it as you want. So a lot of those epigenetic sequencing, they're available in Geo, which we'll cover. Yeah, we can talk about it tomorrow. So those public data, the chipseed data, they're on Geo, so you can just download it, load it onto the genome browser alongside your own RNA-seq or other data set you have. Okay, so with regard to reference genome, I don't know what species organisms you're working with. There are a lot of databases. So one of the most popular are ensemble genomes, and you can see here like different kingdoms, and different species and subspecies, different version for the same species. So it's quite well developed in terms of just how extensive we have sequenced all those different variety of creatures. And then for human and mouse, there are other websites you might find useful, like GeneCode especially. We use a lot of the annotation files from GeneCode because they're better maintained, more extensive. And we will be downloading the annotation data for humans from this website today. And then for mouse, there's another website called the Mouse Genome Informatics. It has more kind of in-depth annotation in case you wanted to see beyond genomes. There are different mutations and then different annotations. There are phenotypes, SNP informations, etc. So you want to kind of integrate your RNA-seq analysis with other readout, you can have a look at this website. And of course, for human mouse comparisons, you can get the orthologs information from here, which I used that before, very handy. Otherwise, you think about do it manually. It's going to be a nightmare. So another thing that you might want to check is the protein database. So going from mRNA to protein is a big leap, but sometimes you might want to kind of check out, like what does it look like in terms of, crystal structures, or for different isoforms of, molecules, how does it relate to the same protein, things like that. Okay, so let's have a look at the files. So yesterday we mentioned that after you align the read to the genome, it will generate a file called the BAN or SAM file. So BAN file stands for the binary equivalent of SAM. So it's actually plain text. So it's like what we saw earlier, like for the read. Basically, you're looking at text file, each row representing a specific information for individual alignment. And you don't really have to get into the details that much because in practice, you seldom really open up individual SAM, or BAM files in terms of what is stored in there. Rather, you use the software to handle it for you. Just make sure that you know what parameter you're using. But sometimes it might be good to check if you really want to get into like specific problems. You want to know that, oh, what is actually in the SAM file, in the BAM file, right? So here is kind of example for us to go through. So there are different columns. So for each row, of course, is individual alignment. And then for each column, there are different kind of names associated with it. So here we have a table trying to explain what individual columns mean. So it can be, the name of that sequence, and then we give a flag, and then where's, coming from in certain chromosomes, where it starts, and then some sort of CGAR score, and then the, specific sequence and the base call quality. So the quality is like what we saw yesterday. So it's kind of a readout of just how good the alignment works instead of the base call. So that's for the read, raw read yesterday. So as I said, a lot of details, but in case you want to check, this might be helpful, might be where you want to have a look first. So just one quick note about the CGAR score. So for alignment, for example, we have a example case at the bottom where we have aligned read to the reference genome, and there's an insertion event highlighted in red with the TGC in between. So this is kind of a tricky case. When you align it, you want to account for potential flexibilities in case there are insertions like this. So what happens is how do we handle it is the CGAR score to indicate potential insertion deletion event. And then, different scores or different CGAR strings or sequences can mean that, for example, we have 100 M, which is 100 matching nucleotide. So it's just perfect match. But for the other cases, for example, here we have 63 match, 3 insertion, 34 match. So that will be the case corresponding to this insertion event where we have 3 additional nucleotide in between. So here are the details in case you want to check for potential alignment variations.
So that was called the CGAR score?
Yeah.
The output of how it matches.
Yeah, exactly. So we have it here. So that's the column in the middle. CGAR.
So what did it tell you again?
It tells you how does it matches, right? So imagine we have 100 base pair read.
Yeah.
If it's a perfect match, it's gonna say 100 M. If, for example, in this specific cases, we have the first 33 perfect match. In the middle, there are 3 insertions, and then the remaining 36 perfect match. That means there is an insertion event in the read, which can happen depending on the sample. Because reference genome is reference genome. I think human reference genome is sequenced from 3 individuals living in Buffalo, New York. Yep. You take their blood, you sequence it, and you example them. And I don't think that represents any kind of human variety in this on this planet. So just imagine each of us have maybe, let's say, 0.5% variation, those SNP in single nucleotide polymorphisms. So the reference genome is just one plain text of 3 billion base pairs. You want to match your tumor sample, your blood sample to this reference genome. Imagine how many, kind of variations there would be, right? I hope that makes the point. So, those variation happens all the time, and you have to kind of keep allowances for it.
So at what point do these variations should be taken into consideration, like the quality of the read?
Right, for mRNA is less of a concern because we are, only really looking into 3% of the genome, which is protein coding. But for a lot of other like DNA sequence matchings, it's very problematic. Sometimes the quality, the alignment code is so poor that you have to use specific variant callers. And those are statistical tools actually to trying to make this inference. And it can comes with a large, much larger percent of uncertainty. So in that case, you're playing basically with probabilities a lot. But for mRNA, I think it's tall. Yeah, I think it's relatively less of a concern. Yeah, for today, so rest assured that, it doesn't really affect us much. But just bear in mind that, when you align something to a reference genome, that comes with a lot of variations of potential insertion, deletion, even like structural changes sometimes. In tumor samples, which is very common, where you have, part of chromosome 1 get, inserted into chromosome 3, and then, losing the whole chunk of, anyway. Okay, so just one more thing about the SAM flags. So we have one column called the flags, and I don't really specialize in this, but in case you want to, find out what they are, you can read up a little bit. So after we have done the alignment, we want to assess, how good they are. So typically, we can align, around like 80% of the read, and that's actually pretty good. And if, it does vary between samples, it's very normal. And then for paired-ended sequencing, they tend to make sure that, the pairs are co-aligned. So that's kind of another layer of assurance. And we can have different quality control tools to go through the band files. And then for today, we want to kind of try alignment of the 2 files that we filtered yesterday. And then that's aligning to the human genome. So we can bring up the same tutorial files that we had tried yesterday. And then we got to look at exercise 4.
Are we talking about KD1 and S?
Yeah, the same file we have dealt with yesterday. Okay, so we have the tutorial files from yesterday. So if you kind of start from the beginning, like we log in again, request a interactive node. So let's get to that. Okay. So we start from exercise 4. Okay, so we can start by finding the same directory we have created yesterday. And then we want to make a new directory so we can start assembling our reference genome. So for many of us, we use, a reference genome all the time. So you want to have some kind of database for that. So for each users, it's good that you index it yourself or index for the lab. So, other people can kind of make sure to use the same version. So today we want to try, let's index a chunk of the human genome together so that you will know how to do that. For the whole human genome, of course, it's going to take a bit longer. So today is more for exercise purposes. And we use these index files to do our sequence alignment.
This is because we know what chromosome CD1 and KD1.
Yeah, so those files are truncated to chromosome, like one part of chromosome 18.
So we are only annotating.
So today we are only like indexing chromosome 18. Right, so 18 is kind of a small chromosome to work with, like usually 1, 2, 3 are the bigger ones.
No. That's a big.
Okay, so basically we want to create a new directory called star database. And then we want to copy the primary assembly file, which is in .fa format. So you can download it and load it to your Hoffman 2 directory, or you can just copy it from the Scratch folder that we have been using yesterday.
Do you want the GR the file to be in star database, right?
Yeah, that's right. Yeah, if you kind of follow it by step, you'll make sure that the files are in the right folder, so that when we assemble them, there won't be path issues.
Yes.
Okay, so hope that went well. For the annotation file, will be downloaded directly from the GeneCode website. So here you pretty much just download the file. So this file is slightly larger, but in the gzip format, I think it's handled pretty well on Hoffman 2. So after we unzip it, it should be around 1.5 gigabyte.
So those are two different commands, right?
Which one?
In that box, WPA is
Yeah, don't forget is just download. Like get it from the web.
After it's on our computer, then we can't see it.
Huh?
After it's done, we can't.
Unzip it, yeah. Yeah. So download might take a couple minutes, right? It should give you a progress bar. It's very handy.
Something is happening.
Yeah, good to have updates. Okay, so after we download it, we will unzip it. And then we'll use these two files to do the genome assembly. Okay. maybe some of us have already unzipped it. Okay, so once we have unzipped it, we can proceed to load the software star. So that's pre-installed, so we can just load it like we did yesterday. And then we want to specify where the FASTA file, the name, and then the GRT file, which is the annotation file. And then just one line code to do the genome assembly, indexing the genome.
So the gen code is the whole genome? The GTF file,
Yeah, that's the whole genome.
and then the FASTA file is just,
It's for chromosome 18.
and so if you wanted to do it all.
We can download the FASTA file from GeneCode or Ensemble. So that's slightly larger. It's around like maybe 3 GB or a bit larger than that. Like we unzip is going to be slightly bigger. So we're thinking of reducing the computational time instead of us waiting here.
Yeah, yeah. And so this is with human, and then.
Yeah, this is very human.
We did human data yesterday, but we also did mouse.
Yeah, we tried the mouse liver data yesterday, and then for the practice for exercise 6, we're gonna do whole thing with the mouse genome. Right, so I have already indexed the mouse genome, so we can just go through it as we have already have the genome files.
Wait, so what line are we on?
So,
What line are we on?
oh, now we are trying to index the genome using star. I hope everybody's with me.
Oh, we run each line separately, right?
It's actually the whole thing. So when you have those slashes, it means that it's just breaking the long command into multiple lines. So I would just copy the whole thing and then paste it in the command line.
How about like the 2 lines before that, like that?
You can even copy the whole thing and then paste it. It doesn't matter. The command line is fast enough to respond. Yeah, just copy the whole thing, paste in the command line. It should be good.
How do you choose the number for the genome assay index?
Right, the basis, I think it's kind of a hyperparameter. So this is what I've been using, but it doesn't really affect the efficiency of it.
But what is it?
What does it do?
specified? Yeah.
I think it's kind of I'll have to look it up. It's been a while. Yeah, let me see. So we can just call star and then put up the help menu. Let's check together. Right, yeah, let it run and I will check the help.
So we're making a database right now, right?
Yeah, we're trying to index the genome. So that's necessary to use the star for alignment. So basically you want to have, it's like have a map of the genome because it's, kind of a linear stream, right? You want to actually make sure that you know where you're looking at, especially imagine instead of doing it in a linear fashion, you can index different regions so that you have different draws. So, when you have sequence coming in, instead of going it through in a linear fashion, you can go through each draw simultaneously. Because when we are doing alignment, we're gonna be using a lot of parallel computing. So instead of searching, every sequence in a linear fashion, you can search in different regions of chromosome A. And for the whole genome, of course, is you can search from, like looking into different chromosomes, different regions of chromosomes. So that's what index is doing. It's actually to make a map of the reference genome we're using, so that instead of doing an exhaustive search, you have a rough idea, like, more kind of trying to match more efficiently. Right? Does it make sense?
This is because it's mRNA, the assumption it's the exons are from the same gene.
Right. So in case there are structural kind of features we can exploit it when matching, that's also going to accelerate things quite a lot.
No, making a genome just organizing the genome.
Yeah, it's like,
Different
make sure that you know where to look into. If
Yeah.
you're more organized, it's
Yeah.
gonna be more efficient.
So good question.
Mhm.
So what's the difference between unzip versus a GU and ZIP? So there are two different unzip zips, right? So there are U and ZIP and this.
Oh,
GU.
GZIP.
Yeah, so yeah, what's the difference?
So it depends on the ending of the file. If it's zipped, you unzip it. If it's gzip, you have to g-unzip it.
Are you deleting any certain files in this case, or
Sorry,
are you deleting certain files in this case?
deleting?
Yeah, like because it's saying deleted it. Oh, wait, I might just misunderstanding it.
Right, so that means once you unzip it, the gzip file would be gone, right? You
Okay.
only have the unzipped file left.
Okay, is that the reason why we are using the GU and ZIP here? So like,
Right, because the original file was gzipped,
oh, okay, GZIP. Oh,
right? So we have to use g-unzip.
oh, that's the, okay,
Yeah, so it's just different tools to
Okay.
compress.
okay.
And then you have to use corresponding decompress.
Oh,
tool, right?
okay.
Does that make sense?
So this is like completely different files. It's
Yeah,
that's not like a
different
zip.
file
It's
format.
not just like a zip file. It's
Right.
kind of gzip. Oh, okay, okay.
Yeah.
That's the reason why. Gotcha. Thank you.
How do we check for? It says once the star has completed, it checks for errors.
Yeah, does it give any output?
Oh, it just says all these things it did. Says I started, the star run, started to generate genome files and finished.
Finished, yeah, finished means it's successful. But sometimes, there's error reading files, and then it will give you error messages on the terminal or in the standard output files. So that would be something that, oh, it didn't really finish. You have to fix the problem and try again. Yeah, because if you're indexing a whole genome, it takes a long time. Like I've tried with mouses an hour. Human maybe it takes more than that. So you want to make sure that you are doing it, with the right files, right? Make sure it's going to be successful. So you want to check the error messages. If there's any, you want to kind of restart, fix the problem.
Is it the next ones? We can use it as our.
Yeah, for your specific, let's say Hoffman, you only needed to index it once.
But we only need it for chromosome 18.
For today's, yeah, it's only chromosome 19. But if you want to, let's say, oh, you have a new organism coming in, and you have to index it first before you can align it, right? But it's one-off practice.
What are we running now?
Are we done with the indexing step? Okay, yeah, so if you are done, you can proceed to exercise 4B, which is the alignment itself. So we'll be using the files that we have processed yesterday. So that if for example, the KD1 or SC. So in this code, in this stretch of script, we are doing it for KD1. So just change it to SC1 for the other files.
So we have to be in the start of this folder.
So we will be in the workshop folder and make a start alignment folder. That's where the alignment BAM file will be stored in.
Oh, okay. We don't have to be in that folder in order to run that line, right?
You want to stay in the main folder
Yeah.
because you want to make sure that you can access the data folder. Right. So what I would recommend is to stay in the workshop folder. Just make a new folder, but not getting into it. And then run the remaining of the code.
That's a fatal error. Exists like
Oh, yeah, just one thing. You wanna. So you wanna go back and one level, so see.
Sorry.
Yeah, you went back to your own direction. Just leaving.
Like if we just.
Yeah, now you're in the home folder. You just go into the worksheet folder. Right, and then you list the files in there. So there's a data folder, but there's a data in data folder, so that's not good. So I would say if you choose data folder, remove RN slash. You want to do folder. And then we try again by creating a data folder. Yeah. So now we just check what's in the data folder. Right, so now it's in the right column. So earlier you had that.
I think I just did it.
Right. Now you can proceed.
Do I need to answer this for?
Don't need to. And you can go through QC. Thank you.
I heard there is a single error. I think it could have been it says to solve the problem. Like this may not. Yeah, you have it, and then I meant the data. That's what gives you the numbers. That GD when GD is gone.
I got page letter too.
Maybe you unzipped it or something.
I think the way I made it, I didn't have a GC at the end because my initial files didn't have a GC.
I see. So you can, you can decrypt it. And try again. Or
Does it have to be zipped up?
This just don't be the GC.
yeah, or you just delete the GC, but somehow you forgot.
And that's weird. I do have the dot, I know. I don't know why. But isn't it allowed? They allow it?
I think that's weird.
Oh, no, unexpected end of file. Yeah, that's finished successfully.
Yeah, sometimes, the file format can be corrupted and that can be issued. So like,
I need to change that. Finish my thing.
You can copy from the scratch. I have the design scratch. Okay. So, view list. For SC, we'll have to maybe in the mostly again.
You don't have that?
I just. Yeah, I will do the
Hey, guys. It's good.
But I can try to do the fonts, and then you can call.
Order, yes.
Let me check. It's a with 8, yes. It's with 8. Yeah.
I shouldn't be in the data folder, I should come out,
Yeah,
be in the.
go to the workshop folder.
Would you normally do this in your home or would you be doing it in Scratch? So I'm.
Depending on how large the data set is, I usually do it in the project folder. So, your research group have a larger storage space. So our lab has 4 terabyte. Because I'm dealing with hundreds of samples. So BAM files, I usually throw it in the Scratch because I don't need it, permanently. Or you can just do it in the project folder. I might occupy, up to 500 gigabyte at one point, but I can delete the intermediate files later. So home directory is only 50 gigabyte storage. So, sometimes it's full and then you cannot do anything about it. Scratch, I think for each of us is 5 or 10
You
terabytes.
said 2 terabytes.
Oh yeah, 2 terabytes. Yeah, pretty good.
So project file folders or not, that's if your lab pays for it.
Yeah, it's a paid storage space and it's shared within the group. So it's permanent and is actively maintained, backed up. Scratch, it will be purged every 3 months now or
2 weeks.
2 weeks.
Yeah.
So, like data in the Scratch folder will disappear.
Yeah, I think our lab doesn't have any, so we have to do it from scratch.
I see. Just make sure you back it up somewhere else.
So you have to index it every time though, the genome?
So I put all of the index in the project folder, so it's, it's gonna be
If you do have a
permanent.
project folder, you're gonna have to do it, index it every time.
Yeah, because it takes a lot of space, isn't it? Like mine takes at least a couple of like 10 gigabytes per genome. Yeah, maybe it's good to have some storage space, like 2 terabytes, not that expensive.
Okay.
And then you can put, those kind of, core files in there. You don't need to actively maintain it.
Thank you.
Just some thoughts.
Can you find the file? And go to the start. Let's just see. So sometimes the indexing didn't really work. Or it wasn't. Check. Yeah, let me see how big that big. Yes. You just leave you.
The map is gonna take a long time.
But the alignment is gonna take a while.
I haven't gotten any activity on my phone.
Right, I did it this morning. I think I just went to do something else. Yeah, I think it was a half an hour or something.
Half an hour, we don't sit here.
We can chat. Maybe it'll take fast, take less than half an hour. Yeah, so you can check out the, like what the code mean. So some of us were asking about the SA index, so I looked it up. So that's one of the parameters. I will put it over here. Yeah, so for each of the softwares, it has a very extensive manuals and, explain what parameter means and what, any guidelines they have for you. So they have this flag, and then it says the default is 14. The input should be integer, and that's the, the length of the SA pre-indexed string. So I think it's a way to kind of decide how big chunk of like nucleotide you are searching in one go. And it makes sense that, the longer it takes more memory. So basically RAM is gonna store the information you're searching for each of those, an index.
Thank you.
And then they recommend that if you have a small genome, you should kind of scale it down. We're using 12 because we're, only dealing with chromosome 18. And we want to make sure to be RAM efficient because we only request a 4 GB of RAM. Yeah, and then there are a couple of other things you can kind of scroll up and down and to see, what kind of parameters you can give. So apparently this flag gives you kind of control of just how memory efficient you are. And also, you're dealing with specific small genomes. You want to make sure that it's done, at a fine resolution.
And this is just for the indexing.
Yeah, it is only for the indexing part. For the alignment, I think we don't have that anymore. Let's move it over here. For the alignment, we have a couple other parameters.
So I guess if the indexing takes more memory, the alignment would be faster.
Oh, those are individual tasks. It has nothing to do with each other. And you're not running in parallel, right? So RAM was more for this specific, instance of computation, how much short-term memory I'm using. And then we are requesting 4 GB. So that's decent for most, kind of moderate intensity computations. But for aligning multiple genomes or multiple samples, you want to do, maybe 4 GB for each job and then scale it up. For single cell analysis, typically you will use 8 GB RAM at least. Sometimes you have to scale it up to like 64. because the computation is more intense.
How much are you allowed to learn? Max?
I don't know about Hoffman too, but I used to kind of run to the ceilings of 96. And then you have to check the individual core. So some core have a bigger RAM. So if you need that much of a short-term memory, you want to make sure you request a specific core has that capacity. And then another option, if you need more RAM, is to go to GPUs instead of the standard, Intel CPUs. So this is very more kind of technical aspects. Yeah, for single cell sequencing analysis, yeah, it does becomes a problem. But for bulk currency, 4 is typically fine. Yeah, sometimes I use 8 for large samples. Like some samples have more depth in reading. Okay, so we're all waiting for alignment, I guess.
It says that it ran.
Are you run?
But I
Great.
don't know, like that was too quick, right? It like ran it in a couple of seconds. I don't know. If
It's finished or it's still?
It's finished successfully.
I see. Then that's great.
Wow, I don't know.
Yeah, it took me a while. I don't know. Maybe the core I got was like just very slow.
How long is that supposed to be rolling? For 20 minutes, 30 minutes?
Well, you were still running, right?
Yeah, it's still
Yeah.
running, so it's normal, right? Or
I think it's normal.
Okay.
Maybe she got a super duper core. You can always check the output. Like, how big the band files is, usually tell, give an idea of how successful.
If I do this, like. Can you, yeah. Log into Hoffman, right? More than once.
Yes.
In parallel.
Yeah.
So maybe I do that on the. because I
Yeah,
think
while
that
you're
I
running on the
see-
internet, you can do nothing, right? So if you really need it, you can open a new window. Yeah, so we have one
two things
here.
to do. One was SC1, one was KD1. So-
Yeah, so here we are on one window. You just open another one and then log in.
Yeah.
Log in
I did it
again.
on to-
Yeah, usually like you can open up several windows, fine.
I've done like 9 where I like like my entire thing will be like 9 different terminals. They're all logged into my husband, but they're on like different screens. So like I literally feel like I'm like the only shift at like some fancy hotel just like like this screen do this screen.
But that's really hard to manage.
Yeah.
I was like, which screen was I at earlier?
The life is still arriving for me. Is that normal?
Yeah, it took a while for me. But that's pretty much like the heavy part of today's exercise. So let's give it some time.
And then I have to leave around 20 minutes. What other exercises are we going over today?
Okay, so if you have one aligned, you can proceed to generating recount table. So that's pretty much the last step of the pipeline. So this is relatively quick. It takes about several minutes to do. Yeah, even I've tried it for 40 samples and it takes about like 20 minutes. So relatively efficient. It's pretty much just counting because you have aligned all the reads to individual part of the genome. Now this step is just counting how many read in this specific gene, or you want to do exons, you can do that as well. So yeah, pretty much just go through exercise 5. It's quite straightforward.
So are we going over exercise 16?
So 6 will be the pipeline, but you can do it at your own at your own pace afterwards. It's fine. So you have to dash out. So the pipelines will kind of get us to try a real sample. So in this case, it's mouse liver sample, Y type and knockout. So yeah, should be good. I tried it earlier. Everything's working. Yeah, so Melanie, that will be maybe for homework today. So for the others, we can kind of wait for the alignment to finish, and feel free to take a break because we're waiting. There's nothing we can do about it. You can open a new console to try on alignment with SC1. So that's another option.
Work, but mine's saying that it finished and then it's like not into a line. So that's why it's going so quick. It's not working.
Oh, it didn't.
No.
Okay.
So I don't know.
Do you want to find out why?
Yep.
Yeah.
What does it say? Like,
So we were just looking at like everything seemed fine.
On. Like this.
started sorting.
And then it's like
I think the input file has an issue.
okay, because I feel like I
Have a
even
look at the input.
so that is being input file is this.
Yeah, it's supposed to be CPC.
Yeah. I mean, I had this file as a fast queue.
Right, what happened?
I had you said to
So there's nothing in it, just. It only had like 37 things.
Right. So is that down to like the fast queue file then not wrong or something like this?
Yeah, I recommended to go through the fast P filtering step. Okay, I just or you can copy the T-zip file from the threshold. So I have that.
Well, so it should be this.
Yeah, so now you want to fix the input. The output from fast P, right? So you can either run that again.
Yeah.
Or you can copy.
I'd rather try to figure out why my codes don't work.
Yeah, let me try.
So, but my file that I'm using isn't.
Oh,
So does that mean I can?
you can just zip it then.
Okay. Do that again.
The G.
I just did the fast key before because I noticed that these ones weren't working. I didn't notice the size.
Yeah, or you can just copy it from my phone.
Yeah, that's fine. Okay. So then now if I do.
Or you can go out one.
Same size. Okay, okay, that's good, that's good. You have to go. Yeah, it should take some. I was like, I know my computer's not super fast, so I can't do that. Thank you. You sure?
So this is the KD one example. Yeah, I think we have the e-signature last time, but it's just an example.
Okay.
Yeah, if you try the liquid data, I think it's gonna be more comfortable.
Is really my problem.
Yeah. And I think it was. Maybe more than that, but maybe more than that.
Yeah. Yes. Yeah, check on the coding sheets.
Okay, we're all good.
Yeah, I'm not frustrated. So I wanted to run the other one, I just run this and with
Yeah, you changed the name.
That's it, right?
Yeah, that's the only thing.
do I save it in a different folder so I can see the
Yeah, you should write out to the star alignment folder.
Oh,
Yeah.
not a different folder?
Why would you put in a different?
Cause it should do it separate.
Oh, so the structure of this code is like all the alignment are in the star alignment folder. So when you are generating the recon table, you can just do it in one go.
Yeah, alright, alright,
I hope the logic
Yeah.
makes sense.
alright.
Okay.
So you have the questions. Also, I'm trying to do this, trying to fast student candidates. Right, so I do have the fast teaching syllabus. In the state of no, we're gonna apply that program as QC, right? So I have the HTML version and the .c. So if
No,
I-
that's not the raw file.
so what should be the file?
So those are the raw files, right? K1.fastq and C1.fastq, there's 3 GB.
So I have to zip that.
You zip, yeah, you zip
it.
Yeah, you can zip it,
Okay.
or you can just copy the zipped version from exercise book.
I thought I didn't have it. Did you make it again? You said you have to run it. You didn't run it yesterday.
I have the raw file, right? That's where you copy it from.
Okay, this is the raw file. See how fast you.
Yeah, and the gzip. Or you can just gzip. Yeah, gzip. That's. No, there.
It's fine.
No, the one people say. Yeah, oh, yeah, you're checking that one.
Yeah, so gzip this guy.
And then it's gonna be gzip, and then you run through the fast P.
It's the one.
You write this compressed.
So the Also, they're the same file, one is David and one is not. You have to always enter the right file. You can't do the operation on something that is not. Did, so we have to pass. Yeah, so that's that one. And then choose it. So now I. Yes.
Okay, have we got to exercise 5 yet? We're still waiting for alignment. Okay. You got to 5?
Yeah.
Okay, all right.
And I should be doing it in the data folder or in the worksheet. They want them to generate, so I didn't think. Once I have the correct file, I should, without weird editing.
Yeah, I think it's done. 1901.
The rest we could, yeah, see. Oh, for a gene terminal, I have to load it.
It's a new session.
I thought it goes into my Huffman account and it's there. I didn't know.
Every single session is in different units. I'll try loading it.
And then I have to make all of things as well.
Yeah, or not, you just load them and then maybe you need the reference.
Because I thought there's.
Sessions. Oh, you don't need that.
Because this is already in the Huffman too. That's what I thought. This is already made.
Yeah, you just slow down. Yeah, you want to do that. Let's start it a bit.
This one. Although there is a star alignment as directory.
Yeah, but you want to specify where the directory is that one.
Take the other one finished.
Yeah,
Just still, it's still the same time. 26. Maybe somebody doesn't there now that they finished because we were all at the same time.
So, you know, when KD1 finish, you can try 5.
Okay. Yes.
So I think all I'm doing is exercise 5.
Are you getting to 5?
No, because I'm still waiting.
Okay.
I'm still waiting.
To finish. Yeah, so you can try exercise 5 later.
Is there any?
You don't, yeah, you don't need the meat to be there.
Yeah, do you lecture now?
Sorry?
Like, do you still, like,
No more lecture.
Sure.
Today it's just going through 5 and 6.
okay,
Yeah, that's
yeah.
it.
Alright. Thank you. Alright.
Yeah, no problem. No, like, sleeping on the 3 months to do this one.
I think so,
Yeah.
yeah, right, yeah. I have to go all over from. Yeah.
Yes, I'm just.
Yeah, I think that's something, oh, that's.
Yeah.
That's why I like, it's finished, but the other one is a start.
Yeah, so it's control.
Oh, no, that's the first one, okay, first one is 26. Yeah.
So that's.
Yeah, but try to still trying to. Yeah, do another.
So now.
Otherwise it will be around a.
So after you're done with that, you go to solo site. So how you do navigate is there is an appendix on. Which. Yeah, so cheat sheet is more navigating in the console.
Okay.
But yeah, how to use non-analysis text editor is separate in the appendix. So move it around, copy and paste, deleting, and then you want to save and exit. So that's what we did. Okay. I think yeah, the file has extra line. It should be- So that's more a batch file. You're supposed to change. This is for the mouse memory. Yeah, don't need that one in there. Right, so now we're back to. And now we're in everything. So you move your first one, I'll be back.
I see. Just change.
And then we can. So just like we're using the raw. And then save by control. And then now you can try running this.
Did you have to create a star database? And I know this. Got that.
So now you have 2 batch jobs submitted. So you can check the status. Using this and then we just change.
That's what the station.
Yeah. So you have 3 injected. Document and then 2 batch job.
Which is this one.
Yeah, the one that you just submitted.
Okay, but it's not there.
It takes a while.
This was the one.
This is a liver sample, I think for half an hour.
Okay. So it's long down, step up, go over there. This is you can, but literally.
Yeah, so for high priority node,
Okay,
that's what we do, but yeah, it's option.
that's. Okay, and then I basically. did
Yeah.
this part.
Now you're.
I didn't know about what you shared.
Yeah, so that's actually separate.
Oh, separate. Okay.
For alignment, yeah, for alignment, we're using the KD1 S1 sample data.
Okay.
So you don't need to wait for delivery. You can proceed to alignment with the KD1.
So I run the cell, this is the one, or do I, yeah, can copy this whole thing, I already have
You check, do you have start on?
was it not copied yet? Oh, it's star database, sorry. So you don't need to see it's here already for list.
We didn't do wrong. Yeah, it's working.
Is this the part that is in?
Yeah, that's the part as well. And then when you're done, you can try exercise 5, which is using the aligned KD1 band files to generate the table. Mhm. Oh, I flashed that out, so you can do one.
Okay, so it's just copying that.
Can you go back here? Yeah, so I hashed it out, so you can see more like that.
Oh, okay.
So those are 2 options, right? So it's with more for like well. But for today, we just have that software.
Okay, got it. Cool, thank you.
Okay, so many of us are already in step 5. So for those who are still waiting for the alignment to finish, we have an option to copy the aligned BAM files to your folder and then proceed to exercise 5, just in case, you wait for too long and there's no point, keep waiting. So for step 5, we want to basically tally how many reads are mapped to specific regions on chromosome 18. And then for this step, we want to have this software in your workshop directory. So that's the software called feature counts. And then that's the one we're using to tally the gene count table. And then for this function, we need the annotation file. So that's the file we downloaded from GeneCode. And then you just specify sample name. And then what it does is specifying what region we're interested in. So we're, tally the exons. And we want to annotate the regions by gene name. So sometimes a lot of the alignments give you entrance ID, which are, just a stream of numbers. But I usually just go with the gene names to make it, more easier to find out what gene that is. So if somebody give you a recon table with all the entrance IDs, you will have to map it to gene ID. So that will be a different, separate step. But if we can specify it in this, in the very first step, then that'll make it easier. So we give the annotation file, and then we specify the name of the output file. So here will be KD1 recon table. And then the input are all the aligned BAM files in the star alignment folder. So if you have done that for KD1, that will be just KD1. If you have done it for KD1 SC1, the final output will be for 2 samples. So they're aggregated in the same table, the same spreadsheet.
For example, if you have multiple samples, you can just do the step 5 once, and then you have a giant table, basically. Right?
How'd you do that, sorry? You've got.
For example, we have done the alignment for both samples, KD1 and SC1,
Yeah.
and they're all stored in the star alignment folder, right? So what we can do is just to do this once for both of them.
So you don't do that next, the sample equals KD1.
Yeah, so sample here is more kind of, a name, give the output a name, but you can use any name, basically. Yeah, this is just a, kind of a placeholder.
So we're gonna run it twice, one with KD1, one with SC1, but it's gonna, the coming up, line of code will merge the two tables together. It just, basically, concatenates the first table that we made for KD1.
Yeah, exactly.
When you, with
Mhm.
SC1 as well, it will be.
Yeah, so the idea is that no matter how many samples you have, once you have done alignment for them, they are all in this, aligned, sorted by coordinate BAM files, and they're in the same star folder. You can just tally them all together. So just run feature count once for all of them. So the outcome, so the result will be a giant table, basically, with specific genes in each row and then samples on each column.
But with, biologically speaking, with where the KD1 and SC1 data sets related to each other to.
They should be
explain-
using the same genome,
Right.
right? We're using the same annotation files. So you cannot combine the mouse and the human data. Yeah, that's about it.
Thinking where they coming from the same experiment, we wanna know what happened. There were different conditions of the same experiment. What was it?
Yeah, it doesn't matter.
Doesn't matter.
I have tried it with, but usually the same experiment were sent for sequencing together. So I tends to, align them and aggregate them together. But you can also do that for different data sets. For example, I have sequenced like mouse samples myself. I've downloaded other people's mouse data. Once I have, aligned them, processed them, I can also tally them together. And then it's gonna be one giant table. Yeah. So yeah, multiple samples, no problem. From different experiment, no problem. Just make sure that they're the same species using the same reference gene.
Yeah, they always the same thing. So this is just an example.
I believe that would be the case.
Yeah, that would be and it's truncated, so I don't know.
Is there like some sort of percentage we give them before?
Right, so I would recommend you try on the mouse and everything, because that's actually the goal.
Okay. Okay,
And
and that's,
then you can compare. For all of the mouse data.
okay,
Yeah, hopefully it's good.
okay, that makes, is that part 6?
Yeah, that's exercise.
Okay, okay,
So
good, I just bring it. It's a big one. Okay.
I do.
I have a big question though. So like, one of them is financial. Yeah, then so thanks, so I just tried talking. This. I switched it. Do you think this is a place where to be?
So for the code, is in another place.
Okay.
1930. So you want that?
Mhm.
Yeah, they just. Okay.
Yeah, one way still.
Oh, is this finished?
Yeah.
Oh, I see, yeah.
I thought
You
I
might be
saw-
complete.
Okay.
That is incomplete. So what you can do is change the star.
Oh, that's the star.
Star
you do
for
for all of them.
Yeah, for all the that.
Okay, got it. Yeah, and then I would just have to do it again with the SE one, when the SE one finishes. Okay,
Oh, this is without the.
oh, so, wait, then.
Oh, cool.
Yes,
So
so,
you can try the one.
and it doesn't matter. Big one. Cool, so it was just. Thank you.
Yeah, that's right.
I'm waiting for.
Oh, yeah, okay.
So someone has tried to generate recon table for both of them. So if you're done with that, you can proceed to exercise 6, which is the entire pipeline. So we can have a quick look at it. So basically we start from the beginning. Gathering the raw data. So in this case, we're using the mouse liver data. So we have 2 samples, 1 wild type, 1 knockout. And it's pair-ended sequencing. So that means for each sample, there are 2 raw read 1 and read 2. So we can make a link file like this. And then we do the basic quality control and alignment. So in this pipeline, you don't do individual steps anymore. So what we have is a script. It says the round star alignment pair-ended sequencing script. So you can copy this script and have a look what's in there. Basically, it does quality control, filtering, and gene alignment in one go. So because the input-output relationship is quite well defined, so we can just make sure that it's basically going through just like a pipe work, right? One end to the other. coming out. So the output will be the BAM files that we have generated earlier. So for the liver sample, if you're doing batch jobs using QSub, it should take about half an hour on the cluster, but you don't need to wait for that anymore because it's a batch job. So once it's completed, you'll get an email notification. So in the meantime, you can do anything else as you like. So after all the alignment has been finished, we can go through the third step, which is the final step, generate the read count table. So we have another bash script called feature counts. And what it does is just tally all the samples that you have. And you're using bash script again to run that. And this is fairly fast. And doesn't matter like how many samples you have. Usually it's like around 15 minutes, half an hour most. So that's about the pipeline. So today we basically, yesterday and today we went through the individual steps. Pretty much like what went in, what you should watch out for, what files are required, and what you should, kind of bear in mind when it comes to practice. But this pipeline is pretty much, hide all the details away so that you can handle multiple samples in an efficient and highly organized manner. So that's about the idea. And we can give it a try. If it's taking too long, you can kind of carry on later or, submit the job and we can kind of head out for today. And you can check the results later today or tomorrow when we come back.
Where's this?
Oh, so this is exercise 6.
Oh, okay.
So if you go back, exercise 6 would have a link to the pipeline, which is a different page, just in case, like it's way too long for you to scroll up and down. Yeah, and for tomorrow, we are doing applications. So that's a different tab as well for exercise 7 to 9. Okay, so feel free to take your kind of own pace and then we can kind of start from here. So it's just super casual from now on. So if you want to stick around, you can. Or if you feel like you have, better things to do, totally feel free.